Hey everybody, I am just getting online with you guys to share some answers and just some thought processes to approach chapter 23 for the quiz on Wednesday. So the review questions that were posted the other day, this is just my walkthrough of those. Before I get into that, I just wanted to say as things get more real and more local to our community and to people that we care about, I just need to keep spreading on to you guys that I care a lot about each of you. And what that means is I'm hoping that you're each doing everything that you can to stay safe, to keep yourself safe, and to keep your families the same, and therefore our community safe. This will continue to impact us in ways that we can't predict and in ways that aren't going to be great at all. And so my thoughts are very much with you guys and your families. I read a quote the other day on Twitter, so I can't even possibly give credit to who the person was. But it was something like, we are not stuck at home, we are safe at home. And if we keep that mindset, as not easy as this may be, I can lean on the fact that by doing this, by staying home, by not getting out and interacting with other people, or interacting with others as little as absolutely possible, I'm keeping myself and my family safe. So keep that in mind, guys, as we continue to move forward. Thank you for all that you guys are doing on your end of things to make things work and to communicate with me and to be proactive by reading through the instructions for assignments and contacting me early versus late in terms of questions or clarifications that you guys may need. All right. So let's hop in here to chapter 23 concept checks first. So I'm in 23.1. And there's all three questions were valid questions. Number one, explain why genetic variation within a population is a prereq for evolution. Remember, that's how we started learning. The very beginning of chapter 22 was talking about what is the raw material, what needs to be in a population to allow for change to happen. And that is frequency. That is the raw material. Without those differences, natural selection can't select for advantageous traits. So therefore, the population wouldn't be able to evolve. Number two, of all the mutations that occur in a population, why do only a small fraction become widespread? So what we want to think about is the combination or the difference between somatic cells and gametic cells. Our gametes are our sperm cells and our egg cells, our sex cells. The rest of the cells of our body, all other 100 trillion or so, are our somatic cells. If a mutation occurs in a somatic cell, that mutation is lost when that individual dies. The only cells that get passed on from one generation to the next and therefore contribute to this understanding of evolution, those are the gametes. So gametes are, if mutations happen in gametes, then those can be passed on to the next generation. But of mutations that do occur in gamete producing cells, many of them don't actually have a phenotypic effect. As we've learned a little bits about protein synthesis, there are long stretches of our DNA that don't code for anything. And there may be mutations that actually happen in coding areas, but they may not actually produce an actual phenotypic change. Finally, other mutations may have a harmful effect that decreases the success of that individual, and therefore they may not get passed on to future generations because that individual may not make it to reproductive maturity. Number three, the final question for concept check 23.1. If a population stopped reproducing sexually but still reproduced asexually, how would its genetic variation be affected over time? Well, we want to clarify sexual reproduction versus asexual reproduction. Sexual reproduction is where you have two separate cells, egg cell meets sperm cell, and you combine genetic information from two different individuals to create that next generation. Asexual reproduction, on the other hand, is just taking one cell, and this is my cell right here. Take that cell, just copy the DNA, and make two daughter cells from that. They're genetically identical to each other and to the original parent cell. And so if this were the case, genetic variation would drop over time. Meiosis, which creates the gametes, involves crossing over and independent assortment of chromosomes, which both contribute to increased variation in the gene pool. Without that process and fertilization, which is the random combination of sperm cells and egg cells, asexual reproduction just doesn't create the same level of diversity/variation. Remember, though, mutation is the driving force for diversity, which all cells will go through. So there is variation created in asexual reproduction, just not on the level of sexual reproduction. All right, let's move down to 23.2, which is our look over Hardy-Weinberg. So concept checks 23.2 numbers 1 and 2 are actually pretty quick questions that can be calculated pretty quickly. If you'll look at my answer sheet when I post this, you'll see the work for 1, 2, and 3 down below. So you'll see my work for question 1, even though I just posted the answers right here. Right down below, this will show you the work on how I got the frequency of big A and small a. For number 2, same idea. These are simple, quick calculations that we should be able to do. And so that's my calculation. You'll see it down here, 23.2, number 2. Directly below that is the one that's going to trip most of you up, but is the one that's truthfully the culmination of our understanding of Hardy-Weinberg. Hardy-Weinberg isn't there just so we can plug letter or numbers in for letters to calculate frequencies of alleles. And that was something that I corrected on some of your work from last week, the Mighty Mouse experiment. I didn't correct by taking points, but I made comments on Schoology that Hardy-Weinberg is not just there so we can test to calculate allele frequencies. It's a test to determine if a population is evolving by seeing if its frequencies of alleles are different than what we would expect. And so as we look at number 3, we're given a situation where we are seeing the exact number of individuals that have all 3 genotypes: big V, big V, big V, big V, big V, little v, and little v, little v. We see exact numbers for each. In this case, when you have exact numbers of all 3 genotypes, you can count the alleles. You can figure out the exact amount of big V alleles there are in this population and the exact amount of little v alleles there are in this population, and therefore we can calculate P and Q that way. So as I go down to this, I want to point some things out. I've gotten my total population calculated up here by adding my 3 different genotype totals. So there's 120 individuals in this population. I'm crossing my fingers I did my math right. And then the total alleles, I'm just multiplying by 2. So if there's 120 individuals in a population, that means there's 240 of the V allele in this population. So I have down below a step-by-step, steps 1, 2, 3, and then 4. Hopefully you guys can read my my attempt at writing semi-neatly. But I've taken you through a step-by-step of how you should do one of these problems. Expect to see a problem like this on Wednesday's quiz. In some way, shape, or form, I will be asking you to know how to do this and to determine if a population is evolving. So if I look at my step-by-step, the first thing I did was calculate the observed frequencies by taking the numbers we were given in the problem. So for instance, if 16 out of the 120 individuals are homozygous dominant, I just type 16 or punch into my calculator 16 divided by 120, and that tells me 0.133. That is my observed P squared. I did the same for the heterozygotes and the same for the homozygous recessives. These are our observed frequencies. Again, this is something that some of you guys got flipped on the Mighty Mouse assignment. Please look at my comments and look at the answer there also. This is what we did first, was calculate the observed frequencies. The next thing to do from there is count the actual alleles, since we have specific numbers of all 3 genotypes. So down below, I figured out exactly how many big V alleles there are, and then divided by the total number of alleles in the population to get P. So if there were 16 homozygous dominant individuals, every single one of them is carrying 2 big Vs. That's why I multiplied by 2. And then here, my 92, those are my heterozygotes. Every one of them is just carrying 1 big V allele, so I multiplied by 1 just to show that. And then I added that together to get 124 total big Vs in this population divided by the 240 total alleles in the population to get P, which would be 0.517 if I'm rounding to 3 decimals. A couple of you guys have asked, how many decimals should we be rounding to? Do not round to less than 2 decimals. If you round to 2, you should be just fine. When we round to 1 decimal or less, your numbers are going to end up being a little bit off and a little bit different than what I'm expecting for your answers. So I tend to go to 3 decimal places, but if you went to 2, you should be fine. So now, taking those allele frequencies, P and Q right here, 0.517 and 0.483, As I say in number 3, use those values to calculate the expected frequencies. That's where we finally do that plug and chug piece that we've worked on first with Hardy-Weinberg calculations. I plug in my P that I calculated up here, 0.517, and square it to get my expected homozygous dominant frequency. So I take that 0.267 and I throw it right up into my expected frequency of my homozygous dominance. I do the same for the heterozygotes and the homozygous recessives, and I put those in here. The question asks, is this population evolving? To determine that, all we do is compare our expected number. Say if I'm looking at my homozygous dominant expected, the expected is that 26.7% of the population should be homozygous dominant, but there actually is only 0.133 or 13.3% of the population is homozygous dominant. That is a significant difference. And for those that may ask, Mr. Dupree, what's actually going to be, how do I know if it's a significant difference? We're not going to go down the road of statistical analysis here, but the numbers that I will give you, you will see a significant difference. It'll be more than 5% difference from the observed to the expected. You will be able to see it. It won't be close if I want you to be able to tell that that population is evolving. If it's not evolving, then our expected and our observed should be very similar for each of the 3 genotypes. So take a look at this. And again, go back to the Mighty Mouse explanation and the answers there. Again, I will want you guys to be able to do this for Wednesday's quiz. Finally, let's see, concept check 23.3. Natural selection is more predictable than genetic drift because that occurs in a non-random way. We can actually look at advantageous traits in an individual and say, yep, that one's probably going to survive better. It has faster muscles. It can run faster and get away from a predator quicker. That one's probably going to survive better. So we can make predictions. But when we talk genetic drift, we have to continue to remember that genetic drift is random. Those are random chance events that can occur to individuals and to populations, so we can't really predict those things. Number 2 wants us to distinguish genetic drift from gene flow. So genetic drift, because they are allele frequencies can fluctuate at random, this process over time tends to decrease the variation of that population. Whereas gene flow with migration, that may bring new alleles into a population, and it may slightly actually increase variation in a population. Your 2 test your understanding questions. Number 1's a pretty quick one. Natural selection changes allele frequencies because some individuals survive. It's all about the survival of the individual that leads to whether or not certain alleles increase or decrease, which leads to the changing of a population. So we start with individuals surviving or not, but we look at it from an evolution perspective. We look at it from the idea that that will change the population. Quick side note, guys, if you have in your textbook these little QR codes, potentially at the test your understanding areas at the end of the chapter or throughout the chapters, if you just take your camera and put it right over that QR code, it'll actually open a link that you don't have to log into, and it will give you more quiz type questions. So that's just another review avenue for you. Finally, guys, number 5. We're given one last Hardy-Weinberg question. A fruit fly population has a gene with 2 alleles, A1 and A2. We don't see if one is dominant and if the other is recessive, but we're just in our minds going to assign P for the frequency of the A1s and Q for the frequency of the A2 alleles. Tests show that 70% of the gametes produced in the population contain that A1 allele. If the population is in Hardy-Weinberg equilibrium, what proportion of the flies carry the 2? So what that's really asking us, what proportion of the flies carry both of those alleles? We want to think in terms of our heterozygotes. A heterozygote is the only individual that has one of each allele, in this case, A1 and A2. So we got to know that by the end, we're actually going to be calculating our heterozygous population. As I wrote down here, the information is basically telling us that P is 0.7, because in the question it says 70% of the gametes produced in the population have A1. So that's 0.7 for P. And then to solve for Q, we would just take our P plus Q equals 1, or 1 minus 0.7 to get our Q of 0.3. Then all we got to do is plug in 2 times P times Q to get 42% or 0.42 for our answer of C. I think that's everything, folks. I hope things are going okay with Schoology, first of all, as I'm filming this Monday morning and it seems to just be intermittently working. Hopefully things get up and running so we can at least just keep progressing through the week. Again, I'll be posting this quiz. It'll be available Wednesday morning by 8 a.m. And you just need to submit by 3:25 p.m. on Thursday afternoon. Again, try not to push it to the very last minute as things can get a little bogged down in the internets for each of our local areas that we're living in. My busy time here in Buckhorn is from like 11 a.m. to 3 p.m. where things just get a little more crowded on the bandwidth. Otherwise, guys, message me or email me if you have questions or need to meet in Google Chat to go over any of these things. Hope you're all doing well, and I will talk to you guys soon. Sorry, no music for you guys. I just kind of got going and frustrated with Schoology, and I didn't even think about playing some music for you. Next time I'll get you. Take care, everybody.